হ্যালো এভরিওয়ান, আমি অনিক সেনা। আজকে আমাদের টপিক হচ্ছে ভাইরাসেস। আজকে ফার্স্টে আমরা দেখব হোয়াট আর ভাইরাসেস। দ্য নেম ভাইরাসেস কেম ফ্রম আ ল্যাটিন ওয়ার্ড ভিরাস which means venom or poisonous fluid। মানে ফার্স্টে যখন ভাইরাস যখন ডিসকভার হয়েছিল তখন মেইনলি ওটাকে বলা হতো ভেনাম বা পয়জনাস ফ্লুইড, মানে বিষ আকারে দেখা হতো আর কি। এটা বলেছিলেন হচ্ছে মার্টিনাস উইলিয়াম বেয়ারনিক। এই যে ইনি হচ্ছেন মার্টিনাস উইলিয়াম বেয়ারনিক, দেখতে পাচ্ছেন। M. Stanley, an organic chemist, observed that virus could be crystallized and consisted largely of proteins. M. Stanley তারপরে discover করলেন যে viruses are mainly proteins, consisting of proteins and can be crystallized. Later, it has been established that virus contains a small amount of RNA or DNA in addition to protein. মানে ভাইরাস কি হলো, তাহলে ভাইরাসে প্রোটিন আছে and RNA or DNA আছে। মানে therefore, virus is an organism of nucleoprotein, and nucleic acid is the agent rather than protein. It means নিউক্লিক অ্যাসিডটা থাকছে, ঠিক আছে, নিউক্লিক অ্যাসিড অ্যান্ড প্রোটিন মিলিয়ে একটা ভাইরাস হচ্ছে, ওটাকে বলছি আমরা নিউক্লিও প্রোটিন। তো হোয়াট আর ভাইরাসেস? ভাইরাসেস আর নিউক্লিও প্রোটিন। ইম্পর্টেন্ট ক্যারেক্টার্স অফ ভাইরাসেস। Viruses are non-cellular, self-replicating agents. They replicate, but they remain dormant when they are outside the host cell. And they are non-cellular. They don't possess any cellular structure. They can grow and multiply intracellularly as an obligate parasite. Obligate parasite means they become living when they are inside a living host, but they are inactive when they are outside the host. It means they can be called inert or non-living when they are outside the host. Depending on the symmetry, there are three types: cubical, helical, and complex. Viruses consist of two parts: the centrally placed nucleic acid that is covered by a protein coat. The nucleic acid is either DNA or RNA, but do not remain together. Remember, guys, the nucleic acid is either DNA or RNA. DNA and RNA both cannot be there together in any virus. The nucleic acid may be single or double stranded. The outer covering, nucleic acid can be single or double stranded, so it consists of many options. Single stranded, DNA and double-stranded DNA, single-stranded RNA and double-stranded RNA. These are the four options that may occur. The outer covering, shell or capsid, is made up of protein units called capsomeres, except some animal viruses which are with additional polysaccharides. So the viruses have an outer covering. Uh, Inside the covering is the nucleic acid, that is, and the covering is made up of protein units, and those are called capsomeres. They have no machinery of their own for protein synthesis. That is why they are inert when they are outside the host cell. So they need the host mechanism for their protein synthesis. During replication of their nucleic acid directs the host cell to make different parts. When their parts assemble together, they form a complete infectious particle. So they use the host mechanism or host cell to replicate their parts which cannot be replicated by them. Uh, this is why they need a host cell to multiply. That is, they are transmitted very easily from one organism to another organism. There are two types of classification of viruses. That is, first is the hierarchical classification of viruses classification system, and the other one is the Baltimore classification system. Firstly, we will learn about the hierarchical classification system. This classification system was discovered by Lyoff in 1962, Lyoff et al., based on the following parameters: the nature of nucleic acid, that is DNA or RNA. So the virus, from the first point, the virus becomes of two types, either DNA virus or RNA virus. The number of strands of nucleic acid, that is double stranded or single stranded. Host range, it means from which distance it can infect the host. Morphology of capsid, that is the symmetry, it is cubical or helical, etc. Presence or absence of envelope, that is the protein coat we are talking about. Here comes the Baltimore classification system. The viruses can be classified into 7 groups, that is classification by genome type, that is the first group, that is can be DNA viruses, a double-stranded DNA virus or single-stranded DNA virus. The RNA viruses can be double-stranded RNA virus or single-stranded RNA virus, which are positive or negative. DNA and RNA reverse transcribing viruses. It means that reverse transcribing means we have heard of transcription, which is the DNA forms RNA. So reverse transcribing means RNA to DNA. So here comes a single-stranded RNA, the virus, and double-stranded DNA virus. Subviral agents, that is viroids, satellites, and prions. It means they are like viruses, but they are not exactly viruses. Here we are talking about the general characters of viruses again. Uh, so the viruses are non-cellular self-replicating agents which are obligate parasites, and here and there they contain either RNA or DNA. We have heard about it in our previous slides. So let us talk about the next point, that is the amount of protein varies from 60 to 90% and the rest is nucleic acid. It means 60 to 90% of the virus is protein and the rest is nucleic acid. A simple virus molecule is often called a virion. They grow and multiply only in living cells. All known viruses are the pathogen of either plants or animals. It means that every virus that we have come across are pathogens. None of them are just existing without being a pathogen. It means they harm either plants or animals or humans. They can exist outside the host cell for long period but remain inert in free state. Again, they become active after entering a living host cell. Uh, it is what I've been talking about is uh, they they become living or active when they come in contact of a living host or a living organism. They can multiply inside that living organism, but they become inactive or inert when they are outside there. They cannot grow or multiply in nutrient medium supplemented with the extract of host cell in which it grows, but can grow only in exact intact living cell. Thus, they are obligate parasite. It means if you prepare some conditions for best conditions for living, uh, like you are giving them food, uh, enzymes, etc., etc., like we maintain bacteria, they cannot live there. They can only live in a living host, in a living intact cell. That is why we are calling them obligate parasite. They are smallest among the infective agents, even smaller than bacteria and varying over a wide range from 18 to 400 nanometer. So we have some examples here. That is parvovirus, which is from 18 to 26 nanometer. Tobacco mosaic virus, that is 17.5 nanometer into 300 nanometer. Smallpox virus is 400 nanometer, and etc. There are different type of shapes. The shapes can be rod, spherical, or cubical. Within the host cell, the virus can grow, but they do not respire, depend on completely on the enzyme system of the host cell for their activity. Now we are going to learn about structure of viruses. The viruses consist of two parts, that is the nucleic acid and the protein coat. So the protein coat is the nucleic acid is inside the protein coat. It means the nucleic acid is covered by the protein coat. So first we are going to learn about the nucleic acid. Viruses can only contain one type of nucleic acid, that is either DNA or RNA. The DNA containing viruses are called deoxyviruses, whereas the RNA are called riboviruses. So there are being two types of viruses, that is DNA viruses or deoxyviruses, whereas RNA viruses or riboviruses. They vary in the structure of their nucleic acid. Most of the plant viruses have RNA, either single TMV or double-stranded rice-rat stunt viruses. That is, single-stranded would be TMV or tobacco mosaic virus, and double-stranded would be rice-rat stunt viruses. Except a few have DNA, either single means single-stranded, that is Gemini viruses, or double-stranded, that is Dahlia mosaic viruses. The animal viruses mostly have double-stranded DNA or either single, that is a single is very rare, that is poliovirus, or double-stranded, that is reovirus. RNA and bacteriophages contain mostly double-stranded DNA, but they also have single-stranded RNA or single-stranded DNA. Each virion contains one molecule of nucleic acid called genome, consisting of nucleotide pairs whose number ranges from 1000 to 2500. This is 250,000. That's 250,000 pairs. Protein coat. The protein coat surrounding the genome is called capsid, and the protein coat together with the nucleic acid is called nucleocapsid. That is what we have been calling the viruses, right? The capsid is made up of large number of protein subunits called capsomeres. So the protein coat that we are calling capsid is made up of small capsomeres which are large in number. Many mammalian viruses have envelope made up of a bilayer lipoprotein, mainly of host cell origin, and that surrounds the nucleocapsid. After the nucleocapsid, some mammalian viruses, which infect mammals, the viruses which infect mammals have another coating outside the protein coat that we are calling an envelope. That is a bilayered lipoprotein for extra protection. Now we are talking about the symmetry of the viruses. The symmetry can be of three types: cubical symmetry, which are calling icosahedral, and helical capsids and complex capsids. First, we are going to learn about cubical or icosahedral capsids. They have polygon with 12 corners. The polygon which they are telling about the head actually. The polygon with 12 corners, 12 sides, 20 sides, and 30 edges. Each side an equilateral triangle. There are two types: pentagons, that is pentagonal capsomeres at the corners, and hexagons, which are hexagonal capsomeres at the center. This is a cubical or icosahedral capsid. This one. Helical capsids. Both nucleic acid and capsomeres are coiled together and form a spiral or helical tube. Both nucleic acid and capsomeres are coiled together and form a helical tube. So capsomeres and the nucleic acid are coiled together, that are intertwined, like this one. All the helical types are RNA viruses and most of them are enveloped. It means that যতটা RNA ভাইরাস আছে পুরোটাই হচ্ছে enveloped. All helical types of RNA viruses are enveloped. Complex capsids, viruses which do not conform to either of the above two types due to complexity of their structure, is called complex capsids. That is pox virus. This one. Viruses have both living and non-living characters. We are going to read about the living characters first and then the non-living characters. All living matter have DNA or RNA, that is nucleic acid, so they have it too. Mutation is well established by the availability of mutant forms in some viruses. It means mutant forms means they can uh, regenerate and they can change themselves through reproduction. They are sensitive to stimulants like radiation, chemical substances. They can multiply in the living cell of the host. The viruses have antigenic property. They can attack specific host. Non-living characters of viruses are the viruses remain inert material outside the host cell. It means outside the host cell, you can say that the virus is non-living. They are autocatalytic in nature. They are devoid of cell membrane and cell wall. They do not contain a cell membrane uh, because they are covered. The nuclear material is covered by capsid, which is a protein coat. The viruses are devoid of cell organelles. They do not possess any cell organelles, just the protein coat and the nucleic acid. The viruses can be crystallized. Now we are going to talk about the tobacco mosaic virus. The tobacco mosaic virus was first discovered by D. Ivanovsky in 1892 because this virus was infecting the tobacco leaves and causing the leaves to mosaic form in that manner. Like they were infecting the leaves and causing the leaves to rot in mosaic forms. It is a rod-shaped structure with a rough surface, looks like a bunch of banana. Each rod is about 300 nanometer long and 17 nanometer in diameter. Each rod consists of two parts, protein coat or capsid and nucleic acid. The capsid consists of 2130 protein subunits called capsomeres. It means the number of capsomeres used in a single tobacco mosaic virus is 2130. The nucleic acid is a single-stranded RNA molecule. The capsomeres are closely arranged and helically around the helix, thereby it follows a hollow cylinder. The RNA remains deeply embedded in the protein subunits 5 nanometer from the outside, thereby it occupies the whole. The RNA thread extends the entire particle in the form of a long helix. The targets of TMV. The TMV particles in tobacco mosaic particles infect the tobacco plants and develop tobacco mosaic virus disease. The virus moves from one host to another by the vectors like Microsiphon solanifoli and Mysis pseudosolani and M. persica. TMV can also infect other solanaceous plants, that is the plants from Solanaceae family like tomato, etc., and also to some other hosts. Uh, don't worry, guys. I will give this slide in the form of PDF in the description. The mechanism of TMV multiplication. The multiplication of TMV in host can be divided into the following steps. Way of entry, that is, the TMV enters the plant body through mechanical wounds by vectors or through infected pollens. Uh, we have just now talked about the vectors, right? The vectors can create wounds, or the wounds that are previously created can be exploited by the vectors. Entry of virus, synthesis of mirror image template copy of viral RNA, and synthesis of new RNAs from the template. After entering the host, the virus becomes active and it starts to uh, think that it requires to uh, regenerate its RNA or reproduce. We can say reproduce its RNA. So it quickly creates a template copy of the viral RNA, and the synthesis of new RNAs are started. Synthesis of viral protein, that is the capsomeres, formation of new virus particle. After the nucleic acid and the protein coat are both created, then the new particles are created, formed. Site of synthesis of viral DNA and proteins are mm, bacteriophages. What are called bacteriophages? The viruses which infect bacteria are called bacteriophages, right? They actually destroy the bacteria. They eat everything within the bacteria. That's why they are called bacteriophages or commonly known as phages. The phages which infect E. coli are known as coliphages. Life cycle of viruses. So the viruses can reproduce by two means or two types of reproduction they have. That is the one first is the lytic cycle, and the second is the lysogenic cycle. So there are quick differences in the simple differences in lytic cycle and lysogenic cycle. We are going to talk about that. In the lytic cycle, the viruses tend to completely destroy the organism, whereas in the lysogenic cycle, the viruses tend to coexist with the organism's DNA. Let's move on and quickly learn about. There is lytic cycle. This type of cycle is seen in T-even phages. It means T2 phage or T4 phages have the lytic cycle, which attack Escherichia coli. The lytic cycle consists of 5 steps. First is the adsorption. There is a simple difference between adsorption and absorption, right? Absorption means which it comes to the when it comes to the inside of something, it is called absorption. Adsorption means sticking to the surface. It means the virus first sticks to the surface of the cell or the cell membrane of the cell. The first specific organelle, the tail, and the receptor site of the host cell is called adsorption. The interaction between them is called adsorption. The adsorption is facilitated by negatively charged carboxyl groups on the host surface and positively charged amino group of protein present in the tip of the tail fiber first attaches to the host surface. Infection. When the adsorption occurs, it means the viruses stick to the surface of the cell membrane, then it will infect. Infection means it will infect with the DNA or the RNA, the nucleic acid it contains. The nucleic acid is going to be transferred from the virus to the organism's cell. After adsorption, the first particle secretes an enzyme which hydrolyzes the murin complex of the host cell wall. The enzyme hydrolyzes the murin complex of the host cell wall, in turn creating a pore. The sheath of the tail then contracts and pushes the central tubular part, the core of the tail, into the host cell like an injection needle. Then the injection needle is pushed and the bacterium receives the nucleic acid of the DNA. That is the infection part. Then comes the synthesis of fudge components in host cell. After infecting the nucleic acid, it will reproduce inside the host cell. Once the fudge nucleic acid takes the entry inside the bacterial cell, it suppresses the synthesis of bacterial protein and directs its synthesize the proteins of the fudge particle. It means after entering the bacterial cell, the viral DNA stops the mechanism of protein synthesis of the bacterial cell completely, and instead it commands the cell to synthesize the proteins of the virus. Formation of new fudge DNA, new fudge particle. The new fudge particles are formed by the assemblage of nucleic acid and protein. The nucleic acid are reproduced in the bacterial cell and the protein as well, and they are combined together, and the new viruses are formed. This is called maturation, which is controlled by viral genome. Liberation of fudge from the host cell. In a cycle of fudge development, about 200 fudges are formed, which take about 30 to 90 minutes in the host cell DNA and secretes lysosome. The lysosome it completely destroys the cell wall, which causes the lysis of the cell wall. As a result, the fudges are liberated. So we have completed the lytic cycle. Then we have come across the lysogenic cycle. In a lysogenic cycle, the fudge is absorbed on the cell wall of the host bacterium and its DNA becomes injected in the host cell, just like the lytic cycle, the first few steps. Here, the fudge DNA, like the lytic cycle, does not take over the protein synthesis machinery of the host cell. Like in lytic cycle, the viral DNA took over the bacterial protein synthesis, and instead it ordered that the proteins of the virus will be synthesized. Instead, here it does not do that. Uh, Here it becomes integrated with the nucleoid of the host genome. It combines with the host genome. The nuclear DNA or nucleic acid of the virus combines with the host genome. This integrated phage DNA is called prophage. It is called prophage when it combines with the bacterial genome. Thus, the new composite genome replicates as one unit. After becoming the new genome, that is the combination of bacteria and virus, it replicates as one unit. The composite genome then multiplies for indefinite number of times and produces daughter lysogenic bacteria. After a number of generations, the viral genome gets detached from the composite genome and releases in the cytoplasm. This dissociation is called induction. It means what occurs is that first the prophage is created, then the prophage multiplies. After a few generations, it means a two, 3, 4, 5 generations after that, the viral genome detaches itself from the bacterial genome. After the detachment, it induces itself in the cytoplasm. This is called induction. After induction, it enters the lytic cycle. It means when a viral genome ordered the bacterial protein synthesis to stop their synthesis and to viruses, that type of cycle, which is the lytic cycle, it enters that phase, and it destroys the host bacterium and the viruses are liberated. Significance of lysogeny. The lysogeny has an important role in the transfer of genetic material from one bacterium to the other. The temperate fudge acts as an agent for gene transfer through process known as specialized transduction. Temperate fudge is the fudge which enters the lysogenic cycle. It is useful in the gene transfer processes, which is called specialized transduction. In this process, when a temperate fudge comes out from the host bacterium as prophage, it may include a part of bacterial DNA along with its DNA by mistake. After lysis, newly developed fudge can infect the new host bacterium, thus transfer the portion of its previous bacterial DNA to the newly infected bacterium. This is how we can transfer new DNA or new nucleic acid. Transmission of viruses. The movement of viruses from one host to the other host is called transmission. This transmission is dependent on the external agents. The transmission of plant viruses takes place by the following means. So, from one host to another, the transmission can be taken place by the vegetative propagation, through collateral hosts, through soil, through seeds, through pollen grains, through fungi, through rubbing and friction, through insects. So there are many ways through which the other hosts can be infected by viruses. Importance of viruses. Viruses are both harmful and useful. Harmful activities. The viruses are responsible for various diseases of both plants and animals. For plants disease, we have tobacco mosaic virus, yellow mosaic of lady's finger, leaf roll of potato, etc. And for animals, we have smallpox, meningitis, pneumonia, mumps, etc. The plant viruses cause damage to different parts like root, leaf, fruit, etc. and cause economic losses by reducing quality and quantity of the plant products. Phages often kill the beneficial microorganism during commercial production of milk and antibiotics. Useful activities of viruses are in space research. Lysogenic phage cultures are used as radiation detector. So for detecting radiation, lysogenic phages are used. Virulent or temperate phages are the phages which enter the lysogenic cycle, help in genetic recombination and are used widely in genetic research. Phages are used as scavengers to eradicate the bacteria present in the polluted water. To a limited extent, phages are used in therapy and prophylaxis of some bacterial diseases. By holding both living and non-living characters, viruses got the importance in determining the origin of life. Viruses can be used to produce vaccines as well, used to develop viral infection. While working with potato spindle tuber disease, T.O. Diner was first to isolate and characterize the causal agent, potato spindle tuber viroid. So we are not talking about the virus right now, we are talking about viroids. So what are viroids? We have learned that the simplest of viruses are called viroids. Uh, these viroids cause the potato spindle tuber disease, that is PTSV. These are smaller than viruses, and he coined the term viroid to those subviral particles. Since then, only 20 plant diseases have been shown to cause by viroids. These are chrysanthemum stunt, chrysanthemum chlorotic mottle, citrus exotis, etc. Only two animal diseases such as crepey disease of sheep and Kuru disease of humans are confirmed to cause by viroids. The viroids are, let's not talk about the structure. The viroids are small, low molecular weight, circular single-stranded RNA molecule containing 250 to 390 bases, which is insufficient to code even for one enzyme that may require to replicate itself, and some even lack AUG, the initiation codon. Viroid do not have the RNA molecule. The molecular weight is around 75,000 to 12,000 Daltons. Now we are going to be talking about the replication of viroid. The mechanism of viroid replication is not yet clearly known. Two hypotheses have been proposed in this aspect or respect. RNA-directed replication, that is, this view suggests that in healthy plants an enzyme replicase is present which accepts the RNA as template, and the RNA polymer is available in the uninfected plant. So RNA polymerase replicates the RNA. DNA-directed replication. According to this hypothesis, the viroid can be replicated on DNA templates. These DNA templates are either synthesized after infection. So these are only hypotheses. It is not something that has been proved or concrete. Viroids differ from viruses. The virus RNA is enclosed in protein coat. The virus RNA is enclosed in protein coat. Uh, the viroids lack any protein coat and the RNA remains free. The size of RNA in viruses is 3.2 to 20 kb. The size of RNA is 25 to 370 bases. 3.2 to 20 kb means kilo base pairs. It means 1 kb means 1000 base pairs. After viroids, we are talking about another subviral particle, that is prions. Prions have been defined as small proteinaceous infectious particles which resist inactivation by procedures that modify nucleic acids. This fact comes as considerable surprise to the scientific community. These diseases are often called spongiform encephalopathies. Prions are macromolecules smaller than any known viruses with molecular weight near 30,000 Dalton. The diseases are characterized by uh, motor control, dementia, paralysis, wasting, and eventually death, typical following pneumonia. Diseases caused by prions. Animal diseases caused by prions are scrapie, sheep disease, PME, transmissible mink encephalopathy, CWD, chronic wasting disease, chronic wasting disease, BAC or bovine spongiform encephalopathy. Bovine means cattle. Human diseases that are caused by this are CID. That is not the CID you're thinking about, it is Creutzfeldt-Jakob disease. GSS, Gerstmann-Straussler-Schrinkner syndrome. FFI, fetal familial insomnia. Kuru, and Alpers syndrome. Thank you everyone for your patience. Thank you for watching this video. I will give this slide as in the form of PDF in my description. So please subscribe and comment if you like, or comment if you have any query. I will try to answer that as quickly as possible. Thank you everyone.